So this is what I wanted to talk to you about today. And I'm going to step back just a little bit and do some background and why I think we can study some aspects of adolescence in an animal model. And it looks like the adolescent may be a fairly highly conserved developmental transition if you define adolescence as the period of time from dependence on one's parents to relative independence. You see very similar traits across a variety of species, not just in humans, but in a variety of species undergoing this developmental transition. It consists of common goals. Basically, you have to learn what you need to survive as an adult and pretty much on your own, and you have to gain the stuff that's going to allow you to reproduce successfully and carry on the next generation. During this period of time, there's similar biological changes. At some point in time during this big, broad span of adolescence, there is puberty, which occurs at some point in time during that period of time. There's a growth spurt. There's other fairly characteristic physiological changes of puberty that you can see across a variety of species. There are also regionally specific brain transformations, which are quite remarkable. Across mammalian species, all mammals contain the same basic brain regions. So you have a ventral striatum in rats, and you have ventral striatum in humans, and ventral striatum in rhesus monkeys, and in your dogs, and so on. Not only are those brain regions highly conserved across species, but the relative timing of when things develop across these brain regions appears to be fairly well conserved as well. You also see some similar age-typical behavioral features during adolescence that you can see in a variety of species. For instance, two that I'll be talking about today are increases in peer-directed social behaviors, and another one is increases in risk-taking. Now, the prototypic, one of the problems with adolescence is you kind of slide into it and slide out of it. And so it consists of a lot of soft signs, so there's no single event or marker that signals its onset or its offset. In humans, for instance, some people often will talk about adolescence being from 10 to 18 years, and now they're talking about late adolescence or emerging adulthood being at this time span between, say, 18 to 25 years, or you could go a little bit over that. And generally speaking, in virtually all species, females tend to slide into adolescence earlier than do males, and males tend to show adolescent-typical characteristics somewhat later. So you'll have some males showing adolescent-typical behavioral characteristics maybe at 25 years or 35 years or 45 years, and so on. The same sort of thing that you see in primates, non-human primates. It varies with the subspecies and the species, but they often refer to it as older juvenile or early subadult, not necessarily calling it adolescence. In our little rodent models, we also can identify periods of time where we see these behavioral changes and these neural changes occurring. In rodents now, I like to talk about early mid-adolescence as from roughly 28 to 42 days. And the late adolescent emerging adulthood is from, say, 42 to 55 days. We don't have a lot of time, so a lot of the tasks that we would like to use in adults that you can leisurely study in adult rats over months, we have to ratchet it down to be able to study in a week or two interval of time. Okay, so the important qualifier is, though, and that's why I put it in there because I'll forget to say this, and it's very important to say that there are many aspects of adolescence that we can't study in animal models. So it depends exactly on what you're interested in studying as to whether an animal model could be appropriate or should be used. So as I'm talking today, you could decide for yourself if you think that the models that we're dealing with may have something to say about human adolescents and their alcohol use. Okay, so I wanted to just really briefly highlight a few of the adolescent brain transformations. This is not really the major focus of today, but we're finding out that the adolescent brain is undergoing fairly marked developmental transformations, and during the last, say, 10 to 15 years, it's been gradually recognized that the magnitude of alterations is fairly large. Some of these changes are regressive, and this includes pruning of the connections or the synaptic connections between neurons, where Pascal Rakesh has shown in non-human primates that in some regions of cortex, up to half of the synapses may be lost during adolescence. It's pretty remarkable synaptic pruning. And this thought that this may be experience-influenced, that's why I put a question mark there, because it's more of a just so story now than the real hard data to show that. But it looks as if, at least very early in life, the same thing happens. But at that point in time, there's this overproduction of synapses, and those synapses that don't make functional connections are culled away. Now, why this is happening again during adolescence isn't clear, but people are thinking that what's happening is that there's a remodeling, so you're getting a much more focused brain, this less plastic and more focused, so you're losing these certain synapses and honing in on connectivity changes that are going to be representative of the adult. This is associated, at least in non-humans, with regionally specific apoptosis, which is programmed cell death. So there's actually loss of some neurons in some brain regions, at least in rat cortex. There's a little bit of evidence the same thing may be happening perhaps in non-human primates, but it really hasn't been studied carefully, and it would be difficult to study in humans. Associated with these regressive changes, at least in part with these regressive changes, there's an actual decrease in the gray matter volume of the brain. Now, the brain is divided into really gray matter regions and white matter regions. Gray matter regions are rich in cell bodies, and their projections, they're dendritic projections. And so it's thought that some people believe that the synaptic pruning and the loss of neurons may actually be part of what's driving gray matter reductions. But it also may be related to increases in white matter, because there's a finite amount of room up in the skull. So if you're going to increase one compartment and you've got gray matter and white matter, you increase white matter, there's also pressure for the gray matter to become more condensed. Now, this white matter volume increases because it's associated with the axons of these neurons that are projecting from one region to another, and these axons become myelinated with a white fatty material that's called myelin. And this myelination process starts very early in life and it continues into adulthood, but there's quite a bit of myelination going on during adolescence, kind of accelerated period of myelination. And what this does is speed dramatically the connectivity, the speed of transferring information down the axons. Connections, the nervous system communicates electrically via these impulses that go down the axon, and also chemically at synapses. And so by speeding up the electrical flow, you're getting synaptic release much more quickly. So to me, I think it's a tremendously mind-boggling thing that you've got these axons becoming myelinated and it's speeding up the transmission across longer brain distances. And that's one big thing that's going on in adolescence. And indeed, if you look at networks of connectivity during adolescence, one of the changes that's happening is things are becoming more strongly connected across longer distances, and you're losing some of the local connections there. This also is thought to be perhaps experience influenced, so that if you've got axons that are very active, they may precipitate more myelination, more thicker myelination, to really speed up their the speed with which they can transmit information. Synapses are really costly, and so is axon electrical flow, because you've got to reestablish the ion flow across the membrane to allow for the next transmission. So when you decrease the number of really expensive synapses and you speed up axon flow and make it more efficient, you decrease the amount of energy needed to run the brain. And indeed, the most costly brain that you have are brains of about 3 to 4-year-old kids. They're really using a lot of their energy that they're taking in is used to run their brain. The amount of energy required to use the brain continues to decline and shows major declines during adolescence to reach the relatively stable and very efficient functioning of the adult brain. All these changes are regionally and system specific. Amongst the brain regions that are undergoing dramatic changes during adolescence are reward and affect relevant subcortical regions, such as the nucleus accumbens, which is also called the ventral striatum, and the amygdala, that if you read this literature at all, you keep hearing about these brain regions that seem to be changing their sensitivity during adolescence. In some respects, it seems like they're becoming hyper reactive and cross-reactive, so that emotions seem to drive, interact not only with the amygdala, but the amygdala will kind of increase the drive to the nucleus accumbens, and so you have these cross-reactions. And then the one area that everybody talks about is that you have these gradually maturing frontal regions of the brain, for example, prefrontal cortex, and that networks involving these brain regions. So it's thought that the reason, the very simple and well oversimplified story is adolescents behave the way they do sometimes because they're more reactive in terms of responding to rewards. They find them particularly reinforcing, and they are more emotionally reactive, and you get this kind of cross-reactivity, and that's countermanded by a relatively immature cortical control, behavioral control system maturing in. And that's well oversimplified. The situation is quite a bit more complex than that in ways that we still have much to learn and much to understand. To give you one example for this is some of my favorite data. This is taken from David Lewis's data. This is a rhesus monkey, I believe, non-human primate. And what's graphed here on the y-axis is the relative density, and it's percent of maximal value. Whenever that maximal value occurred, that's 100%. And then everything else developmentally is reflected relative to that 100% whenever that occurred. And this is the adolescence period. What it shows, this this line here is showing glutaminergic input to the prefrontal cortex. This is the major excitatory neurotransmitter in the brain, and during adolescence there's a loss of glutaminergic drive to the prefrontal cortex. But it's not just excitatory input, there's also a major loss of inhibitory interneurons, in connections with inhibitory interneurons during adolescence as well. This is the input from the neurotransmitter dopamine, which is one of my favorite neurotransmitters. It's very involved in the reward system, has a major projection to nucleus accumbens, has a projection to amygdala and emotional brain regions, and other limbic areas, but it also has a major projection to prefrontal cortex that peaks during adolescence and then drops down dramatically. And just to add another little twist, some things don't change at all during adolescence, and all the interesting action with them is occurring much earlier developmentally. This is cholecystokinin, it's a peptide, and it doesn't change at all during adolescence. Now, all of this is occurring, and I should say, in layer 5, one layer of prefrontal cortex, and you can see all the dramatically different changes that are occurring depending on what neural system, specific neural system you're talking about, even within a really tiny little brain region. This is a summary of a few of the changes that you see. These are forebrain changes in adolescence, and there are changes beyond forebrain, and these are just a few samples. This is actually honestly, it's an old slide. I should really update it. But it makes the point that I want to make, and there's 3 points I want to make, if I can remember all 3. One is that if you look, here you see similar changes occurring across species, again emphasizing that the brains during this period of time are undergoing similar developmental transitions. The second thing I wanted to point out is that you often will see complementary changes occurring in different brain regions, which make it tricky to figure out what's going on. For instance, let's take the dopamine turnover. Here in prefrontal cortex, it's high early and it's followed by a decline, whereas in ventral striatum, it's low early and followed by an increase. So you'll see these kind of complementary changes occurring across brain regions, which let you know that really it's not what any particular brain region is doing during adolescence is important, but it's how they are interrelating to each other. And there's changes in other brain regions as well. Then the third point I want to make is if you look at the players where you're seeing a lot of changes, prefrontal cortex, ventral striatum, amygdala, and other areas of the limbic system, these are brain regions that are known to be very important in risk-taking behavior, social behaviors, rewarding stimuli, self-control. So it would be, given all these changes, it would be amazing indeed if adolescents didn't differ from adults in some aspects of these behaviors. And my argument is that they do. So what are the significance of all these adolescent-typical brain changes? I think there's a bunch of things that matter and are related to these brain changes. One is I think that these brain changes are ultimately occurring to support adult appropriate behavior. So these are the changes, these are the final major changes that are transforming the brain into a mature adult brain. Not that the brain ever stops changing, continues to change throughout life, but this is the change, the last changes that are producing this adult brain. And indeed, you see nice correlations between cognitive and emotional maturation and changes in particular brain regions. Then you also have this hormonal reawakening of puberty, and I think that there's two things going on with hormones and brain. Some of these brain changes clearly must be driving the hormonal reawakening that occurs at puberty. So some of these brain changes are really contributing and driving puberty, and a number of people are studying what specific brain regions are involved in that. But on the other hand, these hormonal increases may themselves feed back on the brain and change the brain functioning. Cheryl Sisk has shown that adolescence appears to be a second organizational period for sexual differentiation of the brain, so that the hormone changes during puberty are feeding back on the brain and changing the brain structures in ways that support, and she's been studying sex-typical behaviors and differences in sex-typical behaviors between males and females. On the other hand, it seems that a lot of brain changes that are occurring at this time are dancing to a different drummer and are not really influenced by hormones as well. And we don't really understand really what is triggering or timing those brain changes. As I kind of alluded to before, people are beginning to think, and it's more of a just so story than fact right now, but early in life we know that the brain is much more plastic than in adulthood, and many people are talking about adolescence as being like the last big plasticity period where the brain can be modulated as it's going into adulthood and as it becomes more efficient. The loss of plasticity, you lose the extra synapses that could have provided new connections, and you're honing in on fewer synapses, and that this plasticity loss is associated with increase in efficiency. But at the same time, it's not like you can put up a under construction sign and have that brain stop working. That brain has got to function to deal with appropriate adolescent behaviors during this period of time. And as many evolutionary biologists will remind us, there's at least as much pressure on developmental processes in terms of evolution as there is in the adult organism. So maybe some of these adolescent-typical behaviors the thinking is, and the argument is, may have evolved to support things that are critical for the adolescent to accomplish during that period of time. So I'm going to just focus here on two adolescent-specific behaviors. One is increases in social interactions and peer affiliations. People have argued that this increase in focus on peers, and for instance, human adolescents spend 4 times more time with their peers than they do adults during this period of time. So there's big focus on peer interactions. And it's been argued that in human adolescents and other species of adolescents, that one of the functions of this is to help individuals develop social skills and support away from the parents and the family unit, and to help guide choice behavior. And in some species, other than humans as well, you see increases in parental-adolescent conflict, particularly very early in adolescence. For instance, in our rats, just to give you an example, during this early mid-adolescent period, we see marked increases in social behavior. One of the questions is, why is there this increase in social behavior? We have a bit of evidence to suggest that adolescents may find social stimuli more reinforcing under situations that adults don't particularly find this stimuli reinforcing. So the way that we look at this is through conditioned place preference, and I'm going to go through it. So this is the apparatus. So basically what we do is we take a rat and we expose them to one side of the apparatus, and then later we'll expose them to the other side of the apparatus. And for looking at conditioned place preference for social stimuli, what we do is we put the rat on one side in the presence of an unfamiliar animal that's same sex, same age peer. And then on the other side, we put it alone. And we do this a number of times, and then we, On test day, we put them in the middle and we open up the dividers between the two sides and we see which side they spend more time on. To the extent that they spend time on the side that was paired with a social stimulus, that's called a condition-place preference, and it suggests that they find that that stimuli is particularly reinforcing. This has been done a lot with drugs, and it works with most drugs, not alcohol so well, but most morphine, heroin, cocaine, amphetamine, methamphetamine. It works, nicotine, it works sort of. It works fairly well. And so we did this for social CPP. Our controls here, and it's important to mention the controls, get equal exposure to both of these sides, and they also get exposed to a social stimulus, but someplace else, another apparatus, another room. So they've had the equivalent experience. They just haven't had that social stimulus paired with one side of this apparatus. So this is, and what we did is we made the animals into two different categories of social wanting, essentially, of social desire. We had animals that either group housed or that were isolate housed animals. Isolate housing is just put them in a cage by themselves, they don't have anything to play with, they're just by themselves, they got food and water. So at group housing, they're with other age mates, usually litter mates, up until the time that they're tested. What's shown here is a coefficient. Basically, the higher the number, the more they liked that side. And we'll be comparing the paired sides versus the controlled side. So if they show condition-place preference, the paired should be higher than the control. So if we look at isolate-housed adolescents and adults, both adolescents and adults, if they're housed by themselves, they find social stimuli really reinforcing. So that the animals that receive paired exposure spend a lot more time on that side than control animals that didn't receive a presence of a social stimulus on that side. And that's not surprising. I mean, they're desperate for social stimulation. In adolescence, you don't really have to socially deprive them. If you test animals that have never been socially isolated, have lived around their litter mates till weaning and then live with their litter mates or other same-sex animals after weaning, they also will show condition-place preference. Under these circumstances, adults will not. So adults will not show condition-place preference unless they've been deprived of social stimulation. Adolescents love it, whether or not they've been deprived of social stimulation. So this suggests that adolescents really find social stimuli particularly reinforcing. They don't have to have been deprived to really find that stimulation important to them. To illustrate is another illustration of how adolescents respond differently to social stimuli than do adults. This is some set of data that we're just now getting ready to submit for publication that to me are really mind-boggling because this is a study where we were basically, looking at the response to a very low dose of alcohol, and we were either looking at that response to alcohol in a social situation. So they were given alcohol and then put for an hour with a social stimulus, same sex, same aged animal. Or they were by themselves. We tested adolescents, we tested adults. We expected to see big effects of alcohol, and the social stimulus might exaggerate those effects, and those effects would be stronger in adolescents. What we found was, not that, story of my life, but what we found was that the presence of the social stimulus, the ethanol dose was too low to see changes in expression. And what we used here is, this is not an fMRI study in rats. Nobody does fMRI in rats. What we're doing is we're looking at expression of CFOS, which is an immediate early gene, which increases in its activity in many brain regions when those brain regions are activated. And what we found is a completely different pattern of brain activation in the presence of social stimuli in adolescence than what we saw in adults. So this was mind-boggling to us. The brain regions that we found, we looked at 15 regions of interest that we had identified beforehand and we wanted to look at based on where we thought we might be seeing ethanol effects. But what we found is these 5 brain regions, basolateral nucleus of the amygdala, central nucleus of the amygdala, bed nucleus of stria terminalis, which is part of the extended amygdala, and then the lateral hypothalamus, lateral septal area, this is another limbic area, where when you tested them in the presence of, when you sacrificed them after an hour in the presence of a social stimulus, you see marked CFOS activation that you don't see when the animals were alone, and we did not see that at all in our adults. As a matter of fact, amongst these 15 regions of interest, there was no brain region where the adults showed greater activation in presence of social stimulus than did the adults tested alone. So we're seeing a markedly different pattern. The social brain of an adolescent is different. This is a social brain of an adolescent, and you don't see that same social brain in an adult. We're also, you see these increases in risk-taking, novelty-seeking, and sensation-seeking. They're slightly different behaviors, but for right now we're going to clump them. And it's been speculated that during adolescence you see increased propensity for this. It's not that adults don't take risks sometimes, but the propensity for engaging in these behaviors is greater during adolescence. And there's been a number of people who have speculated as to what the functions of this increased risk-taking behavior might be evolutionarily. Sheidler and Block have argued that it helps foster peer acceptance in human studies. Wilson and Daly have argued that in humans and in a variety of non-humans, it may enhance the probability of reproductive success, particularly in males. And generally speaking, risk-taking behavior tends to be somewhat greater in males than in females. But a major theory is that this increases in risk-taking, this drive to seek out new stimuli, may help in the process of emigration. And emigration, in virtually every species of mammals, in some avian species actually, either the male adolescents, the females, or both, more typically the males, will leave the home territory around the time of sexual maturation. And it's thought that this helps prevent inbreeding. Because inbreeding depression, or the expression of recessive genes that carry bad stuff, is a big problem. And one way to avoid that is by avoiding inbreeding. And it's thought that this increases in risk-taking could help avoid inbreeding by encouraging emigration. You see these increases in risk-taking and novelty-seeking across species. This is some data of ours from, this is now out, but ours in rats, where early mid-adolescents are showing peaks in novelty-seeking behavior. If you use a subset of the Zuckerman scale, looking at sensation-seeking, this is work by Larry Steinberg, who does really cool stuff. Anyhow, you see, again, these increases peaking in sensation-seeking in early mid-adolescence. We can do the same thing for novelty. Are they just showing, engaging in more interactions with novelty because they find that particularly reinforcing? One way to look at that is with novelty CPP. Not a lot of people have done novelty CPP, but it works basically the same as a social CPP, only every day, rather than putting them in the presence of social stimulus or a drug or something, what we do is we put them in the presence of a new novel thing, like a Pez dispenser and a little ball and whatever. So every day they get exposed to another social stimulus. And we use this very similar design as the previous study I was talking about. So some of the animals were isolate housed, and AKA translated, bored, nothing to do. And the other animals were raised in a social stimulus situation. And what we found in adolescent animals is exactly the same thing we saw with social stimuli. I might add that these are male rats only. We don't see this in female. What we see is that they were showing they were showing novelty CPP, whether or not that they were group housed or isolate housed. In adult animals, very similar to what we saw before. Again, adult males, we see that they showed novelty CPP only when they were isolate housed and hence really deprived of any kind of stimulus, whereas the group housed animals showed no novelty CPP. Again, showing that under certain conditions, adolescents show stronger preference for novelty than do adults. Now, so that's how these brain changes may support adolescent-typical behaviors. And we think that there's important ancillary consequences to this as well. That in addition to, in other words, it probably the brain didn't evolve to change how you respond to alcohol, but with all these brain changes in these regions that are very sensitive to alcohol and are responsible for some of the effects of alcohol, it'd be kind of weird if alcohol sensitivity weren't changed and sensitivity to other drugs wasn't changed during adolescence. If it's the case that adolescent alcohol intake is in part biologically, certainly just in part biological, you would expect to see increased alcohol intake amongst adolescents of other species, and indeed you do. These are some human data from the SAMHSA survey data. These are rat data from our lab, and these are 24-hour intake data. These levels of alcohol intake are about the same as animals that were selectively bred for high alcohol intake, and these are just non-selective, just normal adolescent animals, normal adolescent rats that are showing these very high levels of intake. Well, Adolescents eat a lot and drink a lot too. So the question becomes, is it just that? Are they just drinking more because they drink more of everything else? So what we did a preference study where any numbers above 0 mean that they're preferring the ethanol, any numbers below 0 means that they're avoiding the ethanol. This is a sweetened ethanol solution. By the way, they also drink a lot of non-sweetened solution as well. And this is the sweetener alone, and what you find is that the effect in adolescents, the increased intake, is specific to the sweetened ethanol. You don't see it just with the sweetener alone. So it is alcohol. It does appear that this increased intake is alcohol related. Now, and we've seen increased alcohol intake in a really boring, nauseatingly long list of 2-hour intake tests, 30-minute intake tests, all sorts of different things. And so it's a pretty reliable phenomenon across a variety of different test situations that even in our little adolescent model of a rat, we're seeing increased alcohol intake. The interesting thing is this is related to, at the same time that they're showing this increased alcohol intake, they're showing big changes in how sensitive they are to alcohol. Now, are they more sensitive or less sensitive? Well, yes. It depends on what measure you're looking at. For a few selected effects, adolescents are more sensitive than adults. Adolescents show greater sensitivity to adults to ethanol-induced impairment and long-term potentiation, which is a form of brain plasticity, and more sensitive to alcohol-induced disruptions in spatial memory tasks. Scott was looking at the Morse water maze, for those of you who know that. This data was done mostly by Scott Swartzwelder's group, and they've gone in and looked at LTP not only in the hippocampus, but also LTP and LTD in cortex as well. And they find that adolescents are very sensitive to ethanol-induced disruptions in this form of brain plasticity. We found that adolescents are unusually sensitive to ethanol-induced social facilitation, that their social behavior is enhanced by very low doses of ethanol that don't normally affect adult animals at all. And there's a little bit of evidence. I mean, we have maybe two different kinds of studies showing increases in rewarding effects of ethanol. But that's been kind of an annoying thing to study in rats because rats don't show good CPP for ethanol, which is a long story, and I could talk to you about it at the reception, but later. But it's annoying. But we can use it. And so if we look at heart rate changes and index of the rewarding effects of drugs, we find that adolescents are more sensitive than adults. And we've also seen it with second-order conditioning instead of conditioned place preference because we think conditioned behaviors are interfering with expression of ethanol conditioned place preference. And you can avoid that by going to second-order conditioning. So that's why it's a question mark. We've seen it a couple times, but I would like to be more well convinced. So let's focus a little bit on the social interactions. Now what I want to show you, this is a test apparatus that we use for this. This is a dividing line down the middle is a wall that they can't see through. So there's two apparatuses, one here and one here. There's basically two sides, and there's an opening between the sides so that they can run back and forth. The experimental animal is marked with a mark on its back. The other animal is completely straight off the rack animal that was not treated with alcohol or anything. One of these guys was treated with saline, and the other guy was treated with alcohol. And if I can get this, you'll watch these guys interacting, and so they can run away or run towards the other animal. So this is one of the treated animals. This is another treated animal. And so can you get a feel for which of these guys might have been injected with alcohol? If you're thinking this guy, yeah. So that's the kind of thing that we're seeing with this very intense social interactions that are associated with the very low doses of alcohol. If we go in and look at this, and we're now looking at a very adolescent-typical behavior, which is play fighting, which is a lot of what you saw, where they're jumping on the back of and trying to pin upside down the other animal. That's called play fighting. You see that as you go over higher doses of alcohol, that a very low dose of alcohol, in this case 0.5 gram per kilogram, increases play fighting in adolescents but not adults. Now one could say, well, this is an adolescent-typical behavior. You really wouldn't expect that to be increased. But if you look at social investigation, which for a rat is basically sniffing the anogenital region is where their big social investigation thing. You'll see again ethanol-induced facilitation in the adolescents, but not the adults. So that's now one could say, well, maybe it's just ethanol-induced activation, period. So we did another study where we actually did the same thing, where they were either given a partner or a partner-sized cotton ball, fresh cotton ball each time, so that they could interact with that cotton ball, and they did, and they drug it around and everything else. What you see is indeed, again, the ethanol-induced social facilitation, but you don't see it to other objects. We've done this many different times, and we look at activity, the number of crosses between side to side, and we're not seeing activation just generally, so it's a social facilitation, it's not just facilitation of any old behavior. So that's been very well characterized in our lab, and it is a very reliable phenomenon of adolescence. But other than Scott's stuff with the potentiation and the memory and our social facilitation data and the reward data, for most other things, adolescents appear insensitive, relatively insensitive to adults. Okay, so we see decreases sensitivity to a whole raft of things, sedative effects, social inhibition, aversive effects, motor impairing effects, even hangover effects, even some hangover effects. Adolescents appear to be less sensitive to than to adults. So let me give you some examples of that. I'll start first with the social investigation, because if you guys were paying attention to the previous slides I was showing you, I didn't highlight it there. But if you notice that as you go to higher doses, you get this social inhibition. And indeed, high enough doses of alcohol, you start not seeing so much good social behavior. You can see that in bars as well. That you have trouble interacting socially at the higher concentrations of alcohol. Adults are much more sensitive to that than adolescents. So this is an example. So the equivalent doses, adults and adolescents, you're not seeing the social impairment with the adolescents. If you give a high enough dose, you will. And as a matter of fact, I selected these data. I probably wasn't quite fair. I should have probably selected a set of data that went up a little bit higher, because this is 1 gram per kilogram, about 1.25 gram per kilogram. Under most circumstances, the adolescents will also start showing social impairment. So this need a higher dose to show that. This is looking at ethanol sedative effects. And what we're looking at here is we're giving them a high enough dose of alcohol that they pass out. And then what we do is we put them on their back, and we see how long it takes them to flip over. In other words, to wake up enough, because rats don't like to lay on, back up, belly up, and feet up in the air. So as soon as they become unanesthetized enough, awake enough, they will flip over. So we look and see how long it takes them to recover from different doses of ethanol. These are the doses of ethanol. The adolescents are here, and the adults are here. And you can see that it takes the adults about twice as long to recover from those challenge doses of alcohol than do adolescents. Now, let me point out here that this would be relatively boring. Well, it would be relatively boring to me if it were all just metabolic. Maybe the liver just metabolizes alcohol much more quickly in adolescents than adults. So what we do at the time that they wake up is we look at the brain alcohol levels to see if adolescents are really less sensitive to alcohol, they should wake up when there's more alcohol in their brains. So they should have higher levels of alcohol still in their brains, and yet they can wake up. And indeed, when we do that, what we do is we see that the adults don't wake up until they have less than 270 milligrams of ethanol per 100 gram brain tissue, whereas the adolescents are waking up at higher amounts of alcohol in the brain. So they recover at a time that they have a lot more alcohol still in their brain than do adults. So in other words, these differences across age don't appear to be pharmacokinetic. Okay, so another example of adolescent insensitivity is conditioned taste aversion. And this is a very simple paradigm. Basically, you expose the animals to a novel taste. They taste the taste, and then you inject them with alcohol. If that alcohol has aversive effects, then the next time you give them a chance to drink that taste, to take in some of that taste, they'll avoid the taste. I had a taste aversion to baklava because I went out and had baklava one night, and then I got the flu. So it's a very common thing. And what we did is in the first study, we just used these the doses here in the pink. And what we found is in all the test doses of alcohol that we use, here 0.5, 0.75, and 1 gram per kilogram, adults formed a conditioned taste aversion, whereas adolescents did not show any significant aversion at that dose range. We had to go to a higher dose range, 0.5 and 2.5, to see an aversion developing to alcohol. So we require higher doses of alcohol to develop an alcohol aversion. Now, there's a number of questions that we can ask about this. One is why? Why are these ethanol-typical, adolescent-typical ethanol sensitivities? Why are they more sensitive to certain things? Why are they less sensitive to others? We talked a little bit about pharmacokinetics, and I can say we can eliminate that from studies like I just showed you with the sedative study, but it's annoying in that adolescents do tend to metabolize alcohol a little faster than do adults. Their metabolic rate generally is faster than adults. So in virtually all these studies, we're having to monitor blood or brain alcohol levels and kind of factor that in to make sure that this attenuated sensitivity, for instance, isn't just related to the alcohol. So typically in our studies, well, we're always, and we and others that are doing this work, are looking at the alcohol levels to make sure that it's not just pharmacokinetic, and in virtually all cases it's not to date that we've looked at. We're also very interested in the role of gonadal hormones in this, and that's a whole other side story that if any of you are interested in, I'll talk to you about that at the reception tonight as well. Basically, what with these big hormonal changes occurring at the time of puberty, we're interested in whether some of these changes are being driven by the hormones acting on the brain to change the brain and producing these behaviors. So we've looked at a lot of alcohol sensitivity in animals that have been gonadectomized either pre-pubertally or in adulthood, and looking at all of that, and it hasn't proved to be a major force in terms of alcohol sensitivity, but I could tell you more about that later if you'd like. But where we have seen changes that appear to be relevant to these age-typical alcohol sensitivities is in terms of the chemical systems, chemical communication systems in the brain, the neurotransmitter systems that are synaptically interacting. We've found changes in the glutamate system, the NMDA receptors, and the mu-opiate receptors that are particularly critical for particularly this social facilitation that we've been seeing. And Scott's linked these LTP things to both changes in glutamate and GABA systems. These lessened ethanol sensitivities seem to be related to immature GABA A receptor responses, changes in NR2B glutamate receptors, and also perhaps the kappa receptors in terms of the aversive effects, although the data, we need to collect more data on that point to be sure that we're correct about that. But one of the really interesting things is that adolescents, in part their insensitivity to alcohol is because adolescents show a whole bunch of acute tolerance. And I want to talk about that a little bit because I think this is really intriguing. Basically, their brains adapt really quickly to the presence of alcohol in them. So I'm not going to tell you about that yet. Sorry, I forgot. What I'm going to tell you about, I apologize, got so into is I'm going to talk a little bit about the mu-opiate receptors in the social facilitation. So basically, this is part of the evidence for why we think that the ethanol-induced social facilitation may have a mu-opiate receptor involvement, is that if we go in and if we block opiate receptors and mu-specific opiate receptors, we block the ethanol-induced social facilitation. Here we're seeing this is saline-treated animals given either saline, so these are saline animals, or saline animals were given ethanol, and you see this social facilitation in the adolescents. That social facilitation is blocked by very low doses of naloxone, which is a general opiate antagonist. We see the same thing when we go with a more specific mu antagonist, CTOP, where this is the saline animals, and as you go across increasing doses of CTOP, you're blocking that social facilitation that you're seeing to alcohol. Another way to look at it is that, okay, well, if ethanol-induced social facilitation has a strong mu receptor component, then if we give a mu agonist, we should be able to mimic alcohol, and we should be able to elicit social facilitation. And indeed, this is what we see with Damco as mu agonist. We see a marked facilitation of social behavior in adolescents and not in adults. So again, we're seeing a very similar picture in adolescents in adults with the mu agonist as we do with ethanol. Now I'm going to go to the lesson sensitivity, and I'm going to talk about this acute tolerance. So Melleby, back in 1919, identified and defined acute tolerance. If you look at a injection of alcohol or exposure to alcohol, blood alcohol levels will rise for a while and then they're going to decline. So except at the peak and except when you have nothing in the system, for any blood alcohol concentration, you're going to have that same concentration on the ascending limb and on the descending limb. Definition of acute tolerance is that you're less impaired here than you are here. There's a lot of ways to look at acute tolerance, but basically it's an insensitivity to alcohol that develops within a single alcohol exposure period. So there's lots of ways to look at it, different techniques to look at it, and we've looked at this in many different ways. I'm going to be showing you acute tolerance to ethanol-induced social impairment. And basically what we're doing is we're looking over time at the blood alcohol levels and the decline in blood alcohol levels. And I'll do it this way because so this is time. And the blood alcohol levels go down over time. And then we're comparing that with the amount of impairment. And we're comparing the amount of impairment over the time. If they recover faster than they should relative to the decline in blood alcohol levels, it suggests that they're developing acute tolerance. So it's faster recovery than you should based on what your blood alcohol levels are doing. And what we see here is in adults, these are P70 adults, that the decline in the brain, this is brain alcohol levels actually, over time in brain alcohol levels exactly matches the amount of impairment. We've equated everything to 100% maximum to be able to do these. But when you start out with 100% maximum at 5 minutes and you look at the amount of impairment and then decline in blood alcohol level, brain alcohol levels over time, they're exactly matched in adults. But what starts happening in the older adolescents, the mid-adolescents, and the young adolescents, most dramatic in the youngest adolescents, is they're recovering. Their impairment is going down way much faster than their the brain alcohol levels are going down. So they're recovering at a time when they still have a lot of alcohol in their brains and at a time when adults are still quite impaired. So this suggests that one of the reasons why adolescents are so insensitive to alcohol is their brains are really quickly adapting to the presence of alcohol when the alcohol is on board. Okay, well then the question becomes, and we've been talking about alcohol for a while, but how generalizable is this? And there's a number of ways we can look at the generalizability, because we've been looking at alcohol sensitivity in rats. So how generalizable is it? And there's two ways I want to look at this. First, I want to look at it with regard to other drugs. And I want to take rewarding effects and aversive effects, and I want to look because there's been studies where people have looked at adolescents and adults with other drugs and looked at the reinforcing effects of those drugs and the aversive effects of those drugs. And what they've found is, interestingly, that if you look at the rewarding effects of a variety of different drugs assessed via CPP, because most drugs other than alcohol you can study really well with CPP, what we see is that adolescents are more sensitive to adults to the conditioned place preference to nicotine, to cocaine, and to amphetamine, so that they're more sensitive to the rewarding effects of a variety of drugs, not only for alcohol, as we kind of have seen in some of our studies, but we would like to keep confirming that, but with other drugs, they've seen convincingly, this is some of our work, with a variety of different drugs that you see greater CPP. Not always. Campbell et al. Is one of our studies where we saw no difference between adolescents and adults, but that's not too surprising because there's a very steep dose-response curve on that. We may have been at the wrong dose range. So this seems to be fairly common. It's not only with just alcohol, it's with other drugs as well. What about the aversive effects, where I showed you with the conditioned taste aversion that adolescents show less CTA to alcohol than did adults? We see if you can, when indexed via either conditioned taste aversion or conditioned place aversion, which is just like conditioned place preference, only now you're avoiding the side that was paired with the drug. You can see adolescents are less sensitive to the aversive effects of nicotine and amphetamine. One of the interesting things about these nicotine studies is you can see this within the same study, so that at low doses, adolescents are more sensitive to the positive reinforcing effects of nicotine than are adults, whereas at higher doses, it's harder to get them to show an aversion to nicotine relative to adults. So this general increased and decreased sensitivity seems to go for a variety of other drugs, and it seems to extend to non-drug stimuli as well. And that's kind of beyond the point of what I want to talk to again, but we've looked at taste reactivity for sucrose and quinine, which are ways to measure like liking, the beverage wanting and liking stuff, and we see the same thing with sucrose, with adolescents being more sensitive to intermediate concentrations of sucrose and less sensitive to the aversive taste of quinine than adults. But these are all rats. So the next question becomes, how relevant is these data to human adolescents? And we've been really interested in this question, but of course it's hard to bring adolescents into the lab and give them alcohol. So what we have been doing, we've been doing this work now for a couple of years. I've been collaborating with Steve Lissman, who's a clinical psychologist in our department and also an alcohol educator on campus, on Jerry Johansson. And we've been going to a downtown region of a small town in upstate New York into an area where there are a lot of bars. And this is the bars, 1, 2, 3, 4, 5, 6 there, and there's another one there, there's a huge one there. And we go down, when we do two things. We take down pairs of research associates that go up to people as they're passing between bars. And there's a lot of bar hopping in this area, and a lot of people standing outside. They usually block off the street, standing outside and talking as they're going between bars. And we take these research associates and we ask, and we basically get a lot of survey data on them. We give them the audit. We ask them their motives for drinking, how long they've been drinking, how much they drank, did they drink before they came downtown, did they preload, essentially, and what they've been drinking, all sorts of questions like that, how many drinks they've had. Then a subset of those individuals are invited to go into the tent, this is a test tent. And we set up the tent right there where it says our testing station, and it's divided into two. And what they do is when they go into the tent, we breathalyze them as soon as they walk in. They sit down and we give them a motor test, a cognitive test, memory test, and then we get a DNA sample, and then we re-breathalyze them at the end. And I'm going to show you some of those data a little bit later. What we're really trying to look at now is can we see with cognitive tests and with motor tests the same developmental phenomena that we're seeing in the rats. It's kind of a hassle because we need very large sample size because a majority of the individuals that are there are like 19, 20, and 21, primarily 20 and 21, where we have IRB approval down to 16, and we would like to go up to 30, but I don't know about 30-year-olds hanging out down here. They may not be typical 30-year-olds. So when we get weathered out of this region, okay, upstate New York, right? We're kind of getting to the end of the amount of time that you can test in an unheated tent. We actually have little heaters, but they're not very effective. But if you can't do motor tests, if your hands are frozen. So we will shut down this testing, and then we go to a bar situation where there are older individuals, recognizing that those contexts may themselves have very different effects. Okay, so while we're waiting for those data, are there any other available data out there that we can talk about that might be relevant to what we're seeing in the rats? Actually, if you look at the social facilitation and you read the literature, there's a lot of stuff showing up, things like the literature suggests that most students drink primarily for social purposes. In principal component analyses of high school survey data, the most important factor across all measures of alcohol abuse was drinking for social facilitation. Age distinctions and effects reflect the particular importance of social interaction, drinking occasion in young people. You can keep reading this over and over again. So there does seem this ethanol social facilitation kind of thing going on. What about the lessened sensitivity to ethanol effects? I know of only one study that's really looked at that, and it's a very old study. This was Bihar et al. 1983. And actually, Kenny was the first one that told me about this study. And what they did is they went and gave 18 to 15-year-olds alcohol that produced peak blood alcohol levels about 34 to 35 milligrams percent, which is pretty low. I mean, 80 milligrams percent and above is the NIAAA definition for binge drinking. And they tested them on a bunch of different tests. And these are some of the quotes from their study. In this study of the first intoxicating experience of naive children, little behavioral change was noted clinically, subjectively, or in a validated objective test of intoxication. These children exhibited a smaller behavioral change than expected for their BAL. And that's very important because, of course, there is that metabolism thing. None behaved grossly intoxicated or as adult subjects who all, by the way, have been drinking for a while, right? We were impressed by how little gross behavioral change occurred in the children after a dose of alcohol which had been intoxicating in an adult population. So this suggested that adolescents might be insensitive to these kinds of effects of alcohol, these intoxicating effects of alcohol. And one could say, well, why would it matter if adolescents are relatively insensitive to alcohol intoxication? I mean, wouldn't that be a good thing? It would allow them to drink more without impairment. It may be problematic because this may permit or encourage heavier drinking. Indeed, as I'm sure many of you know, a low initial sensitivity to alcohol is a known risk factor for problematic alcohol involvement, increasing the risk of future alcoholism. And you see this lowered sensitivity to ethanol genetically, to at least certain effects of alcohol genetically in family history positive offspring. Also strains bred for high alcohol preference and intake, particularly mice strains and some rats, also are showing this lowered sensitivity to these kinds of effects of alcohol I've been talking about. Developmentally, you may see this to many of these aversive intoxicating effects that would normally give you feedback that you maybe had enough to drink. In adolescence, and we think that it may be as a function of prior experiences, perhaps developmental tolerance and stressors as well. And so we think that all of these things may add up in individuals that may have come from a family background and they start drinking early and they develop tolerance and they're stressed and they're adolescents anyhow, that all these insensitivities may add up to promote high levels of alcohol use. And indeed, I could take a whole bunch of data from a bunch of different places to talk about the high level of alcohol use during adolescence, but in college drinking populations, but I figured I would show you some of our data. This is from our sample group, which again, its average age is 21 years, and where they're drinking on average 8 drinks. And the blood alcohol levels that they're coming in, a majority of them have blood alcohol levels of greater than 80 milligrams %, or that are in the binge drinking range. And I should point out that these are not exit interviews. These are we're going downtown and starting to collect the data fairly early in the drinking evening. I'm like about 10 o'clock, and we run to about 2 or 3. So we're typically picking up people as they're bar hopping, not when they're going home. And we won't test people that can't stand up, go in and sit down at the thing and take the test, right? So we can only, the highly inebriated, highly intoxicated individuals, we cannot bring into the tent because we can't test them. The other thing I wanted to point out, I think this is the next thing, is the audit scores are coming in, average audit scores are coming in at 12.85. And again, this is what the audit, again, I'm preaching to the choir here, you guys probably all know what the audit is, but it has these 3 subcomponents, intake, dependence, and problems. And anything greater than harmful use is thought to be anything greater than 8. So you have the majority of individuals that are coming in with really high audit scores. Now, I might add that these were intoxicated individuals, right? So who knows what they were saying, right? So the way that we handled that to try to come to grips with that, and the reason why we took, by the way, breathalyzed them twice, is they were then in the tent for about 15 minutes. So if their blood alcohol levels, breathalyzing was high initially because they had just gotten done drinking, they should drop by 15 minutes a bit. Actually, we're seeing really good first test and second test in terms of comparability in terms of the breathalyzing data in most individuals. Because we don't pick up individuals that have a beer in their hand. We won't do the survey with them. So that's been a while since before they get in the tent, because they typically have to wait in line to get into the tent. But they're really anxious to get the blood alcohol levels, and we don't give that to them then. We tell them whether they're in the red, yellow, or green zone, but they say we give them a website and we give them their unique ID number. We tell them if they log onto the website within the next day or two, afternoon the next day, they can get the actual blood alcohol level. And a majority of these individuals do. But when they log on, we say, well, before we get the blood alcohol levels, would you retake the audit? So we got audit data on them when they were sober, and the test-retest reliability is highly correlated. So it does look like they're giving data very similarly when they're intoxicated as to when they're not. So these are suggesting that these individuals are drinking quite a bit and able to be tested under those circumstances. The potential high consequences of this heavy adolescent alcohol use are a lot. We've talked about the acute effects. You've got this, you've got problems with alcohol increasing risk taking, which increases alcohol intake and kind of these feedback systems. There's a disruption in brain plasticity associated with high levels of alcohol use. They're less sensitive to alcohol or taking a lot of alcohol. There may be problems there. Chronic effects, there's a variety of those. There's a particular interest in the lasting neurocognitive consequences and increased susceptibility to later alcohol use disorders. We know the earlier you start drinking, the more likely you are going to have a problem with alcohol in adulthood. But we don't know what that's due to. Is it causal or not? And they're beginning to do longitudinal studies to disentangle that. But we're also in the I'm also part of a major initiative. It's called the NADIO, Neurobiology of Adolescent Drinking in Adulthood Consortium, which is headed by Fulton Cruz down at UNC Chapel Hill. And it's a bunch of research groups, UO1 research groups throughout the country that are looking at lasting neurocognitive consequences of adolescent alcohol exposure. We're looking at adolescent alcohol exposure. Our hypothesis is that it increases social anxiety, and that leads to increased alcohol intake in adulthood. And we're in the process of testing that hypothesis in our rats. So the bottom line is adolescence appears to be a highly conserved period characterized by numerous brain and behavioral similarities, due in part perhaps to these age-related neural alterations. Adolescents show attenuated sensitivity to many effects of alcohol, but greater sensitivity to certain things like ethanol-induced social facilitation and thus disruption in brain plasticity and memory, and perhaps its rewarding effects. And it's sort of interesting, this bias towards enhanced rewarding and attenuated aversive effects is seen with other drugs and other stimuli during adolescence as well in our animal models. So this attenuated sensitivity to ethanol intoxication may permit relatively high levels of alcohol intake among at-risk adolescents, potentially leading to adverse consequences during and outlasting adolescence. So this just acknowledge the people that have actually done this work. I have a fabulous group of graduate students, former and current graduate trainees. I might add Marissa Silveri here was the winner of the 2011 RSA Young Investigator Award, and will be presenting her talk at the next year's RSA meeting. She does really good, cool stuff. She came from my lab and then went to Harvard and just doing fMRI, testing the same things we're looking at. She was looking at GABA systems in adolescent rats, and now she's looking at in adolescent humans. And these are other great students and our collaborators. The ones in green are the ones that are helping out with the human studies, and the ones that are in both do both. So thanks all to them, thanks to the research support, and thanks to all of you for being so patient and listening to me. Thanks. Yes?
Just a quick question about one of your early experiments. It's interesting to think about the factors that could contribute to your conditioned place preference for social interaction experiment. I assume you use novel rats for your conditioning as opposed to cage mates.
Yes.
Is there any chance that adult rats are better at discriminating familiar rats from novel rats as opposed to adolescent?
It's possible. And I am getting more and more interested in social discrimination as just to study that developmentally in adolescence. It is a possibility. Or that, I mean, these are Sprague-Dawley rats, which are like really mild-mannered, but it's always possible that they could have issues like aggression issues with other rats that you wouldn't see in the adolescents, because we never see. Although in our testing, and we look at a lot of social behavior and a lot of adult pairs of Sprague-Dawley rats, we've never seen any aggressive behavior. They're pretty mild-mannered, but that doesn't necessarily mean that they might not be feeling, there might not be some adverse effects of being associated with an unfamiliar adult male if you're an adult male rat. Yes?
This idea about how much people decide to drink, it seems a little paradoxical, because you think that what they're after is the social facilitation, which should happen easily, but then I wonder whether the problem is they don't have any direct feedback about that function, so they have to drink until they're noticeably impaired, noticeable to themselves, so that they know they've had alcohol, or you know, maybe the feedback you got from the, you know, asking people why they drank would shed light on that.
Yeah, yeah, we are looking at drinking motives, and we do see social facilitation showing up. But I think that you know, I don't know. I think it's a really good question. I think part of it is some kids are just drinking for to get wasted, basically, for intoxication purposes, and so that goes beyond the initial. And it may not be so much social facilitation as social anxiety, kind of eliminating social anxiety. And then once your social anxiety is eliminated, then you think you're really charming. You know, I always have thought that a really good public health message might be just videos of guys being really charming when they're not under the influence. It certainly would turn me off. But anyhow, so yeah, but I think that that's a really good point. Yeah, huh? What was the purpose of what? What we're doing with that, what we're really trying to do, and it's just the people that we're testing, since there's so many of the polymorphisms are related to alcohol sensitivity, we're really trying to get at that issue a little bit. I don't know if you know, our sample size is, we just added that component a semester ago, and our sample size on that is progressing pretty slow. So I don't know if that's going to end up being interesting or not, but that was our initial intent, was to look at that. Mm-hmm.
I kind of love your tent methodology. Great. But I'm wondering, I'm wondering whether the types of people who might drink more are the types of people who would have a greater discrepancy between their BAC and their level of incapacitation, or whatever word that you use. So you might be biased almost toward finding the effect of
That we can only, because we can't test the ones that are so intoxicated, you mean?
No, I'm sorry. Because the people the adolescents who drink the most might be the people who have the great discrepancy between blood alcohol and, I don't remember the word.
Yeah, impairment, absolutely. Yeah, I think so. I mean, we're testing the highest blood alcohol levels we have, and we have a number of individuals that are coming in at like 318, 314 milligrams percent. Now, if I had 318 and 304, I'd be coma. You know, I'd be laying out there in the corner. And so we are really biased. And this is a heavy drinking place. There's a lot of kids that wouldn't go here. I mean, you can tell that by the average audit scores coming in at 12 or 13. This is a place where kids go to drink. This is not a place where you go to have a quiet glass of wine with your boyfriend and girlfriend. You know, and that's why we often don't go there on Saturday nights because that's date night. So we'll hit Thursday night and Friday nights sometimes instead of Saturday night, because it's a very context-specific group. I don't know if I answered your question.
It does sound very fun.
I'm always looking for more research assistants.
In the rats, you can experimentally assign, you know.
Oh, absolutely.
To drink, or to have more or less alcohol.
Right.
So you can test the true
Absolutely.
relationship between BAC and.
I see what you're saying, because the person themselves is in control, pretty much, of how much they're drinking. Yeah, absolutely, and that is a really good point. Yeah, so that it is itself is a factor. Yeah, Kenny.
Yeah, you know, it's striking to me how similar the pattern of sensitivities between adolescents and adults is similar to the literature on family history positive and family history negative of perhaps increasing positive sensitivity to reward, particularly ascending limb, and the decreased sensitivity to aversive consequences, particularly descending. And do you think, does that finding with yours perhaps give any insights or hypotheses about family history or genetic vulnerability more generally? There's actually another piece, if I can just add it too, is, Shirley Hill's stuff on P300, showing, again, kind of an adolescent form, but the differences are really marked in adolescence, but they normalize over time. It's more like in a family history positive, at least in terms of those executive functions that are picked up by P300. It's more of a developmental lab than...
Right, right. That's really interesting. And, I don't know enough about the family history positive. I need to go through that literature again. I've just been trying to disentangle from that literature what they're really saying about the rewarding effects, because there seems to be different people saying different things about that.
Actually, your data would map on very similarly, I think, to Dave Newland's differentiator model, where it's basically accentuating both the positive and the negative stuff. I don't know if you've, I'll print it out later.
Great,
But
yeah.
I really think it maps very clearly.
I mean, it's kind of interesting that this enhanced sensitivity to the rewarding and decreased sensitivity to the aversive effects, if you go into human fMRI literature and you look at, like, say, a Cummins response, or a variety of different, but a Cummins response to rewards, you can see the enhanced sensitivity sometimes. It's a long story, but you see that there. But the decreased sensitivity to the aversive effects, you also see in terms of attenuated sensitivity to those aversive consequences, which has led people like Monique Ernst to argue that their adolescents are more, they're less harm avoidant and more reward sensitive, which sounds really a lot like what we're seeing in the rat literature as well.
But it's also asking about trying to find the alcohol rewards. Have you looked at hypersonic mobilizations?
At what? Oh, ultrasound. Oh, now that's a long story. Yes, we've spent a really long time looking at 55 kilohertz USVs, which is supposed to be the giggling, rat giggling, according to Jacques Panksepp. So I have a graduate student that's been doing USVs for years. And the bottom line is that in the situation where adolescents are showing a lot of social interactions, so you deprive them a little bit of social stimuli for maybe 3 hours, and the adolescents can show a lot more social interactions than adults, even without alcohol. You put them in that situation, adolescents are socially interacting more, they USV less, show less 55 kilohertz USV than adults. And it's not that they can't, maybe they can't chew gum and talk at the same, walk at the same time. It's not that they can't ultrasound when they're interacting, because if you isolate house them, which by the way is what Jack Panksepp has studied all the time primarily, is isolate housed animals. If you isolate house them, you ratchet up both the amount way a lot. The USVs are really high, and the play is really high, or the social interactions are really high. So it's not that, but adolescents seem to be showing less of the USV measures that we've used. So then we try to do anticipatory USVs, and the same thing. Adolescents are showing less anticipatory USVs to pending social stimulus, or even to being able to go home relative to adults. So we haven't been able to see that. So now we're doing other kinds of things, and we still have a lot of USV detectors around, trying to understand that, but it's not an easy story. Other questions? You are ready for alcohol disinhibition, right? Well, thank you very much.